So in this presentation, we are going to continue answering biology paper 2 for 2023. We'll continue from question 4 up to question 5. So I did post a video where we answered from question 1 up to question 3. So our question 4 reads, Figure 4 shows the relationship between blood, tissue fluid, cells, and life. So this is the figure showing the relationship between the blood, the tissue fluid, cells, and the life. So question A, identify structures labeled U and T. So here we have the structure labeled R, which is the arteriole where blood is coming in. We have the cells, we have the structure labeled S, we also have the tissue fluid, we also have the part labeled U, and we also have the part labeled T. So question 1, to identify the structure labeled U, this structure labeled U is called the lymphatic vessel. Okay, so this is the lymphatic vessel, and the lymphatic vessel is a vessel that contains valves that helps to transport life. Then here we have the structure labeled T. The name of this structure labeled T is called the venule. Okay, so venules, these are just small blood vessels that carry deoxygenated blood from capillaries and channel it to larger veins. They also help to regulate the blood flow and retain. So this was the answer to question A. We now move on to our question B. So under our question B, we have our question B1. Name one nutrient represented by S. So we can see we have the part labeled S here. What is the name of the nutrient represented by the part S? So here you can have different answers. You can either say the nutrient is amino acids, or you can say the nutrient is glucose. It will score a mark. They will give you a mark. Okay, don't say proteins, because the proteins, they are large, so they can't pass through the walls of the capillaries. So here we can say amino acids, or we can say glucose. It can give us a mark. So this was the answer to our question B1. We now come to our question B2. So under our question B2, compare the blood pressure at R to that of at T and give a reason for your answer. So on this question we are comparing. After comparing, then we are supposed to give a reason for our answer. So we are going to start with the comparing. So at R, I did mention that here we have what we call the arteriole. R, we have the arteriole. Then at T, we have the venule. So the comparison is that the blood pressure at R is very high, while the blood pressure at the venule is very low. That is the comparison. So we have said at R, the blood pressure is higher, while at T, the blood pressure is lower. So we now give a reason. So to give a reason there, why at R the blood pressure is higher and the blood pressure at T is lower. The reason is simple. Here at the arteriole, the arterioles, they have got the narrow lumen, okay? While the venules, they have got the wider lumen. So because the arterioles have got a narrow lumen and the venules have got a larger or wider lumen, it creates a high pressure in the capillaries. Okay, it creates a high pressure in the capillaries, which allows now the nutrients to filter out as the tissue fluids, as you can see right here. Okay, so here our answer, we can say R has narrow lumen while T has wider lumen. This creates a high pressure in the capillaries, which allows nutrients to filter out as the tissue fluid. So that was our answer to this question. Okay, so we can now move on to our question C. So here under our question C, the question reads, explain the role of structure U in relation to blood circulatory system. So they want us to explain the role of structure U, and the structure U, we have said that is the lymphatic vessel. So the lymphatic vessel, at first I did mention that it has got valves that helps to transport life. Okay, So this tells us that it has numerous functions such as fluid balance, absorption, immune function, organic specific functions. So under the fluid balance, the lymphatic vessels, what they do is that they collect excess fluid and proteins from the tissues that cannot return through blood vessels. They transport now this fluid called lymph back into the bloodstream. So under absorption there, the lymphatic vessels in the small intestines, they absorb the fat-soluble vitamins to form a milk fluid that we call the chyle. And this fluid helps to deliver nutrients to the venous blood circulation. Then under the immune system there, the lymphatic vessels, what they do is that they transport lymphocytes, okay, which they transport lymphocytes under the T cells and the B cells to lymph nodes, where these encounter pathogens in the lymph fluids. So now these lymphocytes that they transport, they allow now the immune system to be able to respond to the infections that are affecting the body. So when it comes to the organ specific functions, the lymphatic vessels, what they normally do is that they play a role in immunosurveillance after the pathogens have attacked the body. They also transport the dietary fats and the drainage of the cerebrospinal fluids and the aqueous humor in an organ specific manner. So you can see that there are many roles that are being performed by the structure labeled which is the lymphatic vessel. So in terms of writing our answer, we can say the structure labeled it conducts lymphocytes to blood. It also conducts cholesterol and the fatty acids from the alimentary canal and maintains the fluid balance in the body. So we can say it conducts lymphocytes to blood, it conducts cholesterol and fatty acids from the alimentary canal and maintains fluid balance in the body. So these were the answers to this question C. So we are not done now answering our question 4. We now move on to our next question, which is question 5. So under question 5, we have the first question here. So a hemophilic mother had 2 sons who were both hemophilic. The father was non-hemophilic. Question A, define the term hemophilia. So when you look at the term hemophilia, we can say hemophilia is an inherited disease where a human being bleeds for longer periods than normal due to the poor clotting of blood. So this condition now, it comes as a result of the human being lacking certain protein clotting factors that we call factor 8 and factor 9. Okay, so that is how you can define it, or you can say it is a genetic disease in which blood clots slowly due to absence of blood clotting proteins known as factor 8 and factor 9, leading to prolonged bleeding. So that is what we can say hemophilia is. So here we have said hemophilia is an inherited disease where a human being bleeds for longer periods than normal due to poor clotting of of blood. So we have answered our question A. Let us now move on to the next question. So here we are looking at question B. Using letter X and the small letter there representing the gene which is infected, and the X with a capital letter H representing the normal gene, write down the genotypes of the father. So when you look at the father, this is the male who's having a XY combination. Okay, so here according to the statement, they said the father was non-hemophilic, so this is a normal father who's going to have X with a capital letter H and the Y. So the genotype of the father will be this one. Okay, so what will be the genotype of the mother? According to the statement, the mother was hemophilic, so meaning that this mother was infected with hemophilia. So here the genotype of the female, X with a small letter H, another X with a small letter H. So this will be the genotype of the mother. we have answered our question B. We now move on to our last question, which is question C. Using a genetic diagram, explain why both boys were hemophilic. So here we want to explain why both boys were hemophilic using the genetic diagram. So if you are using the genetic diagram, first thing you need to come up with a parental phenotype. You also identify the parental genotype. Then you write the gametes, you separate them. You write you do the crossing there. After the crossing, then you write the first filia offspring genotype. Then from there you write first filia offspring phenotype. Now this question is carrying 6 marks. So they are saying we need to explain why both boys were hemophilic using the genetic diagram. So after drawing the genetic diagram, then we need to give a statement as well. So let's see how we can answer it. So here we now need to come up with the parental phenotype. So under the parental phenotype, we are looking at a normal male crossed with the hemophilic female. So this will be the parental phenotype. Normal male crossed with the hemophilic female. So you need to put a cross here. After the parental phenotype, then we need to come up with the parental genotype. Okay. So under the parental genotype, for normal male, we said the genotype would be X with capital letter H and the Y. Okay, this is normal. Then for the hemophilic female, the genotype would be X with the small letter H on top. After writing our parental genotype, we need to come up with the gametes. So these gametes are going to separate due to the process of meiosis. So here, these gametes will separate. Then even here, the gametes for female, they will also separate. Then after that, you are going to do the random crossing. So we are going to cross this first gamete with this other one, and the result will give us this one. Okay, then from there we'll cross this one again with this one. So I hope we are seeing how we are crossing, and this will be the results. Then from there we are remaining with this one. So this one will cross with this other one. Okay, and the result will be X small letter H and the Y. Okay, then the same one now, this one also cross with this one, and the result will be X small letter H and the Y. So you can see this that we have, we call this as the first filia offspring genotype. So here we have the F1 offspring genotype. Then from there we need to come up with the first filia offspring phenotype. So phenotype, that is the physical appearance. Okay, and this will be denoted from using the genotype. So we have X with capital letter H, which is showing normal, X with the small letter H. So this is XX normal female. This one again normal female or carrier. Then this one again we have X with small letter H and the Y. So XY is the male. So this is the hemophilic male who was the son. Again, this one again hemophilic son. So we are going to write now our F1 offspring phenotype. So here we have said normal daughter. Again, we have the normal daughter. We can say carrier daughter. Then here we have hemophilic son. Again, we have hemophilic what? Son. So remember, after presenting this, we are going just to be given 5 marks. So we need to come up with a statement. We need to explain why these two sons are hemophilia, why they were hemophilic. So here, the statement now we can give, we can say that both sons, you can see these two sons, they inherited the hemophilic gene, okay, from the mother. So the hemophilic gene from the mother was X with a small letter H. Okay, both sons, you can see they inherited this. Again, they had to inherit again the normal gene, which was non-hemophilic, from the father, which is the Y, right here. So our answer will be both sons inherited the hemophilic gene from the mother and the Y gene from the father. This made them to be hemophilic. So these were the answers to this question. So just on the gametes, a reminder, you can circle them, okay? You can circle them, these gametes, okay, like this nicely, because they separated, okay? So we have come to the end of this presentation. Thank you so much for having time to view this content. This has been your presenter, Mr. Mlenga. Bye-bye.